Hello, today we are presenting the first part of our final project, which is enhancement of enzyme nitrate reductase and its metabolites via the overexpression of NIA gene in Micromonas pusilla CCMP 1545 for its use in saltwater denitrogenation purposes. And our team is conformed by Hania León, María José Briones, Sofía Ruvalcaba, and me, Fernanda Uribe. The UN Sustainable Development Goal number 14 is to conserve and sustainably use the oceans, seas, and marine resources for sustainable development. And it states that the ocean powers the global systems that make the Earth fit for humanity, which means that we need to take care of this critical global resource if we want to have a sustainable future. It also says that marine biodiversity is essential for the health of our planet and its population. There is a significant and constant erosion of coastal waters due to the pollution and acidification of the oceans, and it's causing important consequences in the functioning of ecosystems and biodiversity.
Okay, so nitrogen is essential for many biological processes, but it can also be a limiting nutrient for some organisms. The Center for Microbial Oceanography says that most plants, like algae, need biological available or fixed forms of nitrogen, like ammonia or ammonium, to grow. Nitrogen is the worst pollutant in the world because it damages the oceans, it causes proliferation of harmful algae, it also causes eutrophication and dead zones in oceans, causing that the marine biodiversity is damaged and they cannot defend themselves towards disease. Algal blooms consume dissolved oxygen and they contribute to suffering of marine wildlife, putting it more risks to toxins and diseases. The Ocean River Institute says that nitrogen in blooms produces nitrous acid, which is a greenhouse gas, and it increases the acidity of water, and the oceans absorb more carbon like that. So here we can see the eutrophication process. It's when the water is enriched by nutrients where it has more than it needs, and it causes structural changes to the ecosystems. So the increased production of algae and aquatic plants, and some fishes die. It is mostly caused by cultural eutrophication, in which the water receives more phosphorus and nitrogen than it's needed until it exceeds the capacity of the water body, triggering like this structural changes in the waters. Nitrate is also harmful for aquatic life because it has proven to be toxic, affecting the transport of oxygen in these organisms. The bigger the organism and the more salt the water contains, the less harmful it is. The concentrations of nitrate in the water that has proven to be not that harmful for freshwater animals has been under the level of 10 milligrams in nickel nitrate. And for seawater animals, it can go up to 20 milligrams in nickel nitrate.
The 14th UN Sustainable Development Goal can be solved thanks to genetic engineering because a way of conserving the ocean, seas, and marine resources is via bioremediation, which is the use of microbes to clean up contaminated soil and groundwater. It also stimulates the growth of certain microbes that use contaminants as a source of food and energy. Genetic engineering is used to make microorganisms that can break down pollutants or to boost those types of processes in native organisms with those abilities that help improve bioremediation. In this project, we plan on using the algae's bioremediation functions with its capacity to absorb nitrogen, heavy metals, and other toxicants in aquatic environments to help with the process of denitrogenation of the ocean.
For biological denitrogenation of the water, two main processes need to happen. The first one is nitrification, which requires oxygen, where ammonia is converted first into nitrates and then into nitrates. This is mainly done by bacteria such as Nitrosomonas and nitrobacters. The second one is denitrification, which is anaerobic and converts previously obtained nitrates and nitrates into nitrogen gas. This is done by a variety of organisms. Several enzymes and genes are involved in these two processes. For nitrification, the main enzyme is ammonia monoxygenase, codified by the AMO-A gene in Nitrosomonas. Hydroxylamine oxidoreductase also participates. For denitrification, the main enzymes are nitrate and nitrate reductases, but others might be involved. Examples of genes codifying for these two enzymes are NITA and NARG. For the purpose of our research, Micromonas pusilla was selected. This is a marine green algae found mostly in England. This organism has an NIA gene, which codifies for nitrate reductase. Therefore, our focus process became denitrification. Here is the nitrogen metabolism pathway. In red, the denitrification process is shown, and the participation of the nitrate reductase in this is circled in blue. This might help in tracing the pathway for denitrogenation of the ocean. Speaking about previous research, there has been studies about removing nitrogen from water. In this, a consortia of algae and bacteria was used. Bacteria use the most oxygen for nitrification, so algae can provide it to them, while digesting compounds at the same time. Algae solo system had as main factor their medium source. When coming from secondary effluent, an efficacy of 99% was obtained. The consortia systems, when being microalgae assisted, meaning algae provide oxygen for the bacteria, had an efficacy of 85 to 99%. However, when being microalgae dominant, meaning that algae make the digestion, 78% efficacy was obtained. The conclusions are therefore that algae systems are useful and low energy, and the suggestions involve using algae that produce nitrate reductase. The second study is similar, however, in this a consortia of algae mix was used. The main concern was proving this consortia might have a prolonged use, so cryopreservation was applied to it. Algae in the consortia were selected due to their nitrate reductases. The fresh algae had an efficacy of 99%. However, with a higher concentration, more enzymes were present and the nitrogen uptake was quicker. The cryopreserved mix had a similar efficacy of 98 to 99%, meaning that their properties were not affected.
A last year study from the Wageningen Institute of Animal Science in Netherlands explains the effectiveness of integrating an algal reactor in recirculating aquaculture systems by testing the nitrogen removal rate it performed and the economic feasibility. The objective is the recycling of resources in the medium without damage, having as result a nitrogen removal rate average of 1.4 grams of nitrogen per square meter per day. The main conclusion is that for the future, science needs to improve technological advancements in algae culture, such as the design of the reactors or developing low-cost elements. A fourth investigation related to this topic was made this year about coral and algae dominance, denitrification, and fixation capacity. In this study, the turf algae and hard coral took the spotlight of the investigators because of their high-level capacity to fix and denitrificate the nitrogen present in the ocean. Turf algae, once again, is considered a pioneer for ecological practices, and the goal of biotechnology in this topic is to understand the nitrogen activity and determine a boundary in its biodisponibility. So for this genetic engineering project, the objectives are to understand the use of algae accumulation, contribute to the 14th SDG in a biotechnological way, and suggest a methodology for using the advantages of algae. Being more specific in this point, we want to improve the overexpression of the NIA gene from Micromonas pusilla, responsible of the production of nitrate reductase enzyme that has important metabolites for the denitrogenation purposes. Thanks for your attention. Here are our references.